morning, everyone. Thank you so much to the MLCB organizers. I'm really honored to be here today. So today I will be talking about some of the work that we've been doing with self-supervised deep learning to understand big biological image data. So this is what my data looks like. So these are high-counter screens that collect images of proteins for most of the proteome for various organisms. And these screens try to collect systematic data for as many proteins as possible. So each image is tagged with, each protein is tagged with 3 fluorescent proteins. So the green in these images shows the localization and expression of a particular protein, okay? And each image will show a different protein. So in terms of the questions you'd ask using data sets like these, you may be interested in figuring out what proteins are similar to a protein of interest in your data set, right? Because that often implies similarities in function. Or you may want to ask how proteins change localization or expression between different cancer cell lines, because that tells us something about regulation under cancer, right? So to answer questions like these, you would typically have to compare hundreds of thousands or millions of images. That is a number too large and too subjective for us to look at by eye, so we really need computational methods to deal with data like this. So for us to enable computational analyses, we would like to convert our images into representations or feature vectors of measurements of the biology in our images. And what having a good representation does for us is that it really makes analyses and computation a lot easier, right? We can do classification or clustering on the representation basis then. So the question then is, well, how do we convert our images into a good representation that captures biological information within these microscopy images? So if we're talking about representation then, I think deep learning method is usually understood to give you the best performing representations. But there is a catch. For us to get good representations, you usually need a lot of labeled training data for the biology that you want to study. I have a table here of how different representations do of classifying the type of drug images of breast cancer cells have been treated with. And you can see that while a deep learning model trained with a lot of labeled data seems to do very, very well and better than that classic features designed by human experts, deep learning methods that are trained in an unsupervised fashion without labeled data don't seem to do quite as well. Now, this is a problem, right? Because if you look at the scale of labeled data that you need, you typically need tens of thousands, if not millions of images. So that takes a lot of time and is a lot of work. And this problem really bottlenecks our ability to get useful representations for our data sets through deep learning. So this is also the problem that I'm going to address today. Can we find a way to give our models good representations of biology, but without the cost of needing a lot of labeled training data? So to address this question, we started looking at recent insights in what is called self-supervised learning. And the idea is that self-supervised learning trains neural networks on puzzles where training labels can be defined automatically without human intervention. So one great example of a self-supervised method is shown here. So it asks the model to predict how much an image has been rotated by. So rotating an image is something a computer can do by its own, right? So you don't need a human in the loop to generate training data. So the model is trying to predict how much an image is rotated by, and then something remarkable happens. If you look at the representation that is learned by this model, it actually is very good at classifying the type of an object, such as if it was a dog or a car or an airplane in an image, and almost as good as if you trained the model directly. So why does this happen? So to understand this, we'll look at this image over here. You guys can tell it's obviously being flipped upside down, right? And that's because you see that there are human beings in this image, and those people have been flipped upside down. But that requires an understanding of the natural world, right? You need to know what people are and how they're normally orientated, right? So the model has to develop this understanding to become really good at the task, and that's why it becomes good at detecting objects then. So we were very inspired by that method, but we couldn't apply it out of the box. So microscopy images are rotation invariant, right? Instead, we decided to come up with our own method, and we call it paired cell impacting. So the idea is that this is an image of human cells. So for many experiments, you image the same protein as expressed in genetically identical cells under the same condition. So a lot of the time, not always, but enough of the time that it's an okay assumption, the cells will have a similar level of protein expression. Most cells will. So the idea then is that we will take one cell from the image, and we're going to show that to the model. We're going to take another cell from the image, and we'll show that to the model, but we're only going to show the model the shape of the cell, shown by channels like DAPI or tubulin. And then we will ask the model to create a synthetic image of what it thinks protein expression looks like in the second cell. So what is a slow and good representation of protein biology? Suppose I gave you these two images, and I said that the protein biology in these images should be pretty similar. And then I asked you to draw a picture. Well, you would solve this by looking at this image and going where the protein, the green, is expressed in the nucleolus of the cell. And then you would know to draw a bunch of bright green dots in the nucleus of the second cell, right? But that relies upon looking at this image and understanding the protein biology in that image, so we think the model is going to have to do the same thing then. So that's the method. Now I want to show you how it actually works at learning representations of protein biology. So the first data set we started off with is called the Cyclops data set, and the major aim of this data set is to label proteins by their subcellular localization. So these classes that you see down here. And to assist in the development of automated methods, the curators have produced a nice set of over 30,000 labeled images for 17 classes. So we can use this labeled data to benchmark how good a representation is compared to others. So we trained an unsupervised model using one of the unlabeled experiments from no database, and then we benchmarked it upon the supervised labeled dataset then. So here are the accuracies for various classifiers using our feature representations compared to others. And you can see that our method significantly outperforms other methods in classifying protein localization in single-use cells, regardless if you're building a linear or non-linear classifier. And the computer methods include extensively engineered features that have been designed by bioimaging experts, as well as other unsupervised deep learning approaches. And remarkably, you can see we come very close to the performance of the state of our supervised method, and this is in spite of us not using any labels for protein biology whatsoever. So the next thing we wanted to assess was, well, how general is this method? So we took two other high-throughput data sets that are very different from the Cyclops data set. So one of these data sets is Imogen Brightfield, and the other uses an unusual nuclear pore marker. So one thing to note about these data sets is that they have never been analyzed automatically before, only through laborious manual inspection of tens of thousands of images. There's no labeled training data associated with each data set, so supervised learning isn't possible until you get that. And there are properties that make it challenging for more traditional methods. There's a lot of overlapping and out-of-focus cells in this data set, and this data set has no cytoplasmic markers, which is required for many types of feature calculations. So here are what the representations of the proteins for each data set look like. I've reduced them using UMAP, and I've colored them using previous manual labels. So you can see that for all three data sets, distinct protein localizations are well separated. And the separation for the two new data sets is about as good as that for the first data set, which we showed you that the features are very good for them. And hopefully I've convinced you of the performance of these representations, because the next thing I want to kind of move into a little bit is some of the applications that we have been pursuing with our representation then. So these are representations for human proteins that we've trained and extracted using the Human Protein Atlas. In total, there are about 12,000 proteins represented in this clustered heat map over here. So we've clustered them, and then what that does is that groups together proteins with similar representations. And then you can see, as expected, there are very strong enrichments for Go annotations for cellular component within each cluster. So we've been applying these kinds of clustering solutions to find rare cases and then find subclasses of protein biology. And one example that I'll give you today is proteins that localize to the nucleolar rim. So they look like this. It's a ring around the nucleolus of the cell, okay? So the Human Protein Atlas only documents one example, and then we decided to see if we could find more. So we looked at the one example that they gave us, and we found that within about the 30 closest proteins, we found 13 new examples of nucleolar rim proteins, some of which are shown here. And for these proteins, some of them are transcription factors. The nucleolar rim acts as a site of storage until they're required. So for example, for differentiation. So findings like these actually help us create novel hypotheses. In this case, it helps us hypothesize about the regulatory mechanisms of some of these proteins then. So in conclusion, I introduced a new self-supervised method for learning representations of multi-channel microscopy images. It produces high-quality representations for analyses of protein biology with no feature engineering and no manually annotated data sets. And the representations can be purposed for a variety of analyses, from classification to exploratory analyses to more specific questions. You can look at the paper if you're interested in what else you did with the representations. So thank you to my lab, but also thank you to our collaborators who provided the data sources. So you can understand this is a project that used a lot of different data, and we could not have done it without people being open and willing to share the data with us. So if you're interested, you can check out the paper. There is a GitHub link in the paper, and I've done my best to make the model as accessible as possible, but if you have any problems or anything, please contact me. But that's all I have for you for now then. Yeah, oh, way in the back, okay. Mm-hmm. Yes. Mm-hmm.
Do they make errors in the same areas?
You mean the confusion matrices, or?
Yes.
Okay, that's a good question. We did not check that specifically. I think one of the reasons why is that although we did build a classifier, we were actually not really interested in building like the best possible performing classifier. It was really just more like to prove what we could do with the classification, right? And to understand that, I think that, I mean, we think that, like, at least from my perspective, I think that, like, it's true that, like, within this field, it's very popular for people to build classifiers, but we often have to understand that we're operating with, we're biologists, right? We often operate with incomplete knowledge, or, like, we don't know our classes, or maybe sometimes we have, like, misconceptions about our classes, like, for example, like, we think, like, what is one class is actually two subclasses, or something like that, right? So I think that although the classification metric was a good proxy for us, we didn't want to kind of like rely too heavily upon it just because we don't really think it reflects underlying biology, right? But what we do know, however, is that, so like what's really interesting is we think that the biases from classification data set, because they reflect human biases, tend to influence supervised classifiers in a way that they don't influence self-supervised classifiers. And to show you some data for this, let me see. Great. So these are some of the classes as visual in our labeled classification data set, and then you can see how they form clusters, okay? So these are representations from supervised representation, and you can see that each of these four classes tends to form a single distinct cluster. Whereas with the self-supervised representation, you can see that they actually start forming like multiple clusters for each class, even though the clusters are still like adjacent to each other. And what we've been analyzing is that we can actually observe that some of these subclasses seem to be biologically relevant. So for example, within the periphery subclass, which was this blue one over here previously, the two clusters seem to correspond to smooth-looking cell periphery proteins versus bumpy cell periphery proteins. So I think that this is actually a really important conceptual point. Although we often train these classifiers supervised, we have to talk about what biases we're introducing to a representation that reflect our prior knowledge, right? The reason we like these self-supervised methods a lot is because that they kind of teach themselves, and in that way, it's unbiased by prior knowledge, and then you can kind of discover things that would be hidden by your preconceptions of your data otherwise done. I mean, that was a little bit off tangent, but hopefully it answers your question then. Mm-hmm.
Thank you.
Yeah, okay.